Welcome to our plant editing session. I think everybody enjoyed Dan's keynote speech, and in his speech, Dan set the stage very nicely to give an overview and the state of the art in plant genome editing field. I'm not sure anyone knows this. Actually, this year, 2020, marks the 10-year anniversary of the first gene-edited plants developed. 10 years ago, 3 groups, including Dan's group, demonstrated the gene editing plants in Arabidopsis and corn using magnuclears and the zinc finger nuclears. Since then, technology has evolved rapidly. Now we're entered through the Talon technology, and now we're at CRISPR age. In the past few years, the CRISPR-based tools expanding so rapidly, and now we are at the point in plant community, we have the capacity to create many different sophisticated forms of edits, not just knockouts, and in many different plant species at a much higher efficiency and with much more precision. So looking forward, in next decades, I think we can anticipate to see the continuous wave of technology breakthroughs that can undoubtedly accelerate our basic and translational research for plant communities and also lead to many novel agriculture applications and products. So today's session, we invite 3 speakers to cover all these frontiers from both academia and industry sectors. Our first speaker is Nat Graham. He from Pairwise Plants. Nat received his PhD from Jim Birchler Lab at University of Missouri and then completed his postdoc training at the Voitas Lab. In 2018, he joined Pairwise Plants, which is one of the leading plant genome editing companies focusing on crop, vegetable, and fruit improvements. And he will talk about their efforts in Pairwise to advance the editing technology and develop new food varieties. Our second speaker is Heidi Kaeppler. Heidi from University of Wisconsin-Madison, where Heidi is currently a faculty member in the Department of Agronomy and also faculty lead of the Wisconsin Crop Innovation Center. One of Heidi's research areas focusing on breaking the major technology barriers for genome editing, plant genome editing, such as improving plant transformation, regeneration, and delivery of gene editing reagents. So today he will talk about his recent work using nanoparticles to achieve genome editing in plants. Lastly, we have Dr. Bing Yang from University of Missouri, where he holds a joint position as a full professor in the Division of Plant Sciences and a member and a PI for Danforth Plant Science Center at St. Louis. Bing has been a pioneer in the plant genome editing field since the Talon days. His research focuses on developing genome editing technologies in a number of plants, particularly crop plants, and leverage them to study plant and microbial interactions. So today he will talk about his work in genome editing in that directions. Without further ado, I will turn over this to Nat.
Should be good. Okay, so thank you so much for the introduction. It's really an honor to be here, especially coming after Dan, where I did my postdoc. It seems like just a second ago, but it was almost a couple of years now. So I am a molecular biology scientist at Pairwise. So if you haven't heard of Pairwise before, we're a fairly new company we're a food company. So we're in the applied space for genome editing. And so just because of the format and kind of the short time, I can't really go into as much detail as I might like on anything. So what I figured I would do is kind of give you guys an insight into what you need to be able to build out a tool portfolio for food company, and kind of what makes it different when you start doing editing for plants versus when we therapeutics, which is what a lot of people are familiar with. So pairwise, we are basically are founding around the problem of food. And most people are generally familiar with thinking about food of people don't get enough food. That's generally how it's pitched, but it turns out that it's just as big of an issue of the type of food that people are getting. It turns out that 65% of the world is in a country that actually has more deaths due to obesity or being overweight than lack of calories. And in fact, even in the United States, the type of food that people are getting, like 9% is the number of people that are actually getting the recommended amount of fruits and vegetables that they're supposed to be getting. And this is an interesting problem because if you look at it, a lot of people, not everyone, but a lot of people have access to healthy foods, but they choose not to eat them. And so they choose when they're in the grocery store, and instead of going for the fruits and vegetables, they'll go down to the snack aisle, right? A lot of us, myself included, will do this as well. And it turns out as you start asking people why that's the case, that it comes down to a lot of cases to convenience. And so people don't find things like fruits and vegetables to be as convenient to eat. And so they'll go for the bag of chips. So our main goal here is really to focus in the consumer space around making the foods that we can get in the produce aisle a little bit more convenient. So cherries and berries, which have pits or seeds that are often people find very annoying, removing those sorts of traits that can make it so that people are more likely to reach for them. And then also at the same time, making so these types of foods are available year round. So you're not reaching for a thing of blackberries and getting it all mushy. And so our main focus right now in the beginning is pitless cherries and seedless blackberries. And then also we're in the space a little bit with leafy greens. So it turns out if you're making a salad with something like romaine, most people are doing the kind of a lettuce that's not very high in nutrition, but has less flavor because things like kale are really pungent. So things like removing pungency. And so as we are looking at these sorts of fruits and vegetables, we realized that this is kind of a barrier for consumers, but there's been there's not been very much innovation over time for these sorts of crops. And so if you look at it from a historical perspective, over time, back in like the 30s, we were pretty much in the same spot between corn and cherries, for instance, where they were harvested by hand and with a very low yield. But the technological advantages have really been applied to the row crops like corn and soy, where now they're doing a production harvesting that's really automated with a high yield advantage. Whereas specialty crops, consumer crops as we often call them, have been kind of left behind. And so while there's a little bit increase in yield, they're still harvested the same way. And so we were really thinking that with the advent of gene editing, that we could actually have the next revolution in agriculture, and that this is going to be able to apply to not only just the row crops, but the specialty crops as well. So that was kind of the impetus in starting the company. And so it was founded kind of on that idea. So you can see, here's our founders on the right here, of who we have three main academic founders, Feng Zhang, David Liu, and Feng Zhang, who obviously are big names in the gene editing field. And then Tom Adams and Haven Baker are kind of our business leaders that came in as well to start the company. With that kind of really broad base of technology and also leadership experience, we kind of have a pretty strong foundation to build a company out. So we have pretty large, wide base of tools available to us through licensing. We're expanding that and patenting along the way. And through a partnership with part of our funding is through a research collaboration with Bayer, we have access to a transformation pipeline that's kind of state of the art, which allows us to apply these tools. So as we started out in the editing space, what we quickly realized is a lot of these tools are really powerful, but a lot of them are built for therapeutics, because that's generally where they're generally developed at. And so, which is great, but the therapeutic applications aren't necessarily going for the same end result that we would want to in plants. So in therapeutics, when you're often looking for disease-causing mutations and resolving those back, you're looking for something with really high target specificity and a high product purity, okay? And that is good, and that's something that you really need to be able to do in a therapeutic space. But in plants, where you're dealing with something with a really wide range of standing variation already, introducing more diversity is also something that you want. And so we're looking for tools that can increase diversity, both phenotypically and at a sequence level. And then we also share some of the same types of goals that you do for making tools for therapeutics, something where you need a lot of high target density, high PAM density for target accessibility. And then we also need to do a wide range of editing types. So we share some goals, but there's some things that are different. And so this is kind of where we lead off to when we think about developing new tools at Pairwise. So I kind of go through three main things that we kind of think about. The difference in genome complexity, how we have to deal with a different transformation process, and then our diversity pool. A little bit more detail, but I'm not going to talk about it today, but also realize that when we talk about plants, that we're dealing with something that grows in a wide range of temperatures. And so as we apply tools to plants, something to keep in mind is that we're not able to just grow them at 37 where the enzymes are generally developed for. So it's something to keep in mind as you're thinking about applying tools to plant systems. Okay, so let's talk a little bit about what you have to do for making tools work in a complex genome. And it's helpful here because the general solution to this is the same as in therapeutics. You need something that can do high activity at the same time as getting a lot of PAM availability. And if you're not familiar with plants, to kind of give you an example of what genomes in plants are like, here's an example of brassicas on the left here. So if you think about something like mustard greens or leafy greens, these are actually results of two diploid species kind of in a hybridization event making a tetraploid. And that's not all that uncommon. Things like duplication events are not uncommon in the plant kingdom. So over time, triplication and duplication events are pretty rampant over plant history. And so if you look at this from an editing perspective, where maybe in this gene family wanted to knock out this gene, early on you may have 1 or 2 copies you may need to deal with. But now, because of the duplication, triplication events, there could be 50 copies that you need to deal with at any one time, which means you need to have a really highly efficient editor. And you need to be able to have one that can also access a lot of different PAMs to be able to do that sort of editing strategy. So that's really where we started when we started developing systems for plants. And so if we kind of look at what we did, we really just started with what is often overlooked, I think, and when you're developing tools into plants, with just optimizing the expression level, getting them to work higher, and that's a lot of things, promoter, terminator, codon optimization, everything, to make it so that your tool can function at a high rate. And so as we look at it for Cas12a, we were pretty happy with the ability to do that. So if we compare it to just previously published target, we're just by doing some optimizations, we're able to do better than published. But we're really excited about it when we apply those sorts of strategies to Cas9 base editing, where previously it looked like it was not as efficient. It was not a very easy strategy to do in plants. We're able to, through just optimization alone, improve the activity to be as good, if not better, than Cas9, which is something that we can then use to be more precise with our editing strategies. And we're able to at least be, have that in our toolbox for in a high rate. As it turns out, you can apply these same types of optimization strategies to the berries and consumer crops. So here's an example of some of our early experiments in caneberry, where we're able to start developing callus, and we can find editing as a result of that. And so we're able to see where we're able to find editing strategies up to 50% in our caneberries through similar strategies. And what this allows you to then do, as we start looking at enzymes that previously weren't shown to not function in plants very well, is we're able to now kind of rescue those enzymes and make them work a little bit better. And so things like Cas12a from the AS variant, which was shown previously to not work in plants, we can get to levels that are acceptable for a trait pipeline. And this is really exciting to us because this allows us to expand our accessibility. So as we all know, Cas12a from the LB variant generally used in plants has a TTTV PAM, which is fairly restrictive. We'd love to be able to get to a place like Cas9, where if you look at all the different Cas9 variants and put them together, you get about a 1 in 4 PAM density, every 1 in every 4 base pair kind of PAM density when you look at them all together. And it varies depending on different crops because of GC content and things like that. But now as we add in ENAS-CPF1, and as well as some other variants that are possible with Cas12a, when you combine them together, you're able to actually approach a PAM density that's very similar, if not better, than Cas9, depending on the crop that you're looking at. And so by just kind of looking and making sure that you're optimizing expression strategies, we're able to utilize tools that previously were shown not to function as well. So another thing to think about from a plant perspective and editing is the transformation process. And so, kind of Dan alluded to in the previous talk, is that it's not very easy to transform plants. It's a very difficult process that generally uses bacterium. And the process is also restrictive if you're trying to develop tools, because we want to make tools and iteratively test them and improve them. And that's not something that you can then do in the target genome that you're actually going to be working in. And so what this means is you need to have a model system in order to do an iterative testing strategy in any decent amount of time. And so we've kind of built out a discovery platform at Pairwise that's kind of focused on doing a rapid turnaround cycle. And so it allows us to go from design and then building out different reagents. So whether that be doing in-house protein purification or vector synthesis in QC, and then testing in different platforms. We do have and develop some proprietary testing strategies within plants that are very quick, but we've actually started focusing more so on the human cell platform, which is something that's fairly unique in ag biotech companies. But we found that by just taking the human cell facility, there's a lot of advantages, whether it be just the ability to transform reagents at a high rate, or just being able to do a high iterative strategy that really gives us a really high advantage there for developing tools, even though they're specifically for plants. And so over time, we started to use these systems pretty highly. We've developed some proprietary base editors, diversifiers, and things, as well as getting high efficiency targets and base editing strategies from just using this type of a pipeline. I'm gonna give you an example of kind of what this can kind of do for us. I'll give you a couple examples that are in various stages of development. So we're highly focused on Cas12a or CPF1, and we wanted to be able to apply the base editing strategy to that system as well. This is something that was previously published, but it was really pretty low efficiency. We have a really, really good protein sciences group at Pairwise, and so they looked at Cas12a and designed some various linker and or structural architecture designs based on Cas12a and the cytosine base editor. And through our testing program, we were then able to quickly take a large amount of the designs that they were able to push out and test them pretty quickly. And what we found is that compared to like a non-optimized kind of control, that we're able to find architectures that are actually better than what we expected and are fairly high in activity. And so this is really exciting for us. And so now we're in the process of continually checking on the activity of those, both in more human cell testing and in plants. And so there'll be more to come from that soon. We also like these human cell facility because we're able to test different types of delivery architectures that are complex and more difficult in an easier delivery system. And then we can learn if it's something that's of interest to us before we have to worry about optimizing it for plant expression. So one thing you probably have heard of before is a SUNTAG system that relies on attaching a peptide epitope to a protein of interest and then recruiting another protein to it by using an antibody tag. So this is something that's generally done for protein labeling. It's also been done for base editing with Cas9 before. And so we wanted to apply this to Cas12a to see if we could have a high rate of base editing through this strategy as well. And so in the human cell facility then, as a lot of people in therapeutics know, but is kind of new to us in plants, we're able to then deliver all these reagents separately in a high efficiency manner. And so what we've learned is that you can indeed do a SUNTAG kind of base editing approach with Cas12a at a fairly high efficiency. In fact, by with recruitment and having a high local concentration of the deaminase, we're able to get a fairly good rate of base editing with Cas12a. Additionally, another thing that we're working on in the base editing space is continually looking at other types of deaminases that could potentially have a higher amount of editing activity. The one that's been published before to show activity is A3A. And so we're able to, through our experience, we've also seen that that does work in our hands better as well. So whether that be in the top panels here, you can see comparisons through SUNTAG or not SUNTAG with different deaminases. And we see that the A3A deaminase does consistently work better overall. And when we just take it, thunderstorm here, if you do take this into a CPF1, not necessarily the linker optimized CPF1, but just standard CPF1, and we apply different deaminases, we can get base editing with them, but it is always consistently higher with A3A. And so in our hands as well, we find that the A3A deaminase does tend to have a higher rate of activity. Okay, and so in the last couple minutes, I just did want to go over some of the advancements we've made in looking at diversification. So this is something that is fairly unique, we think, in plants, that most people that are generally thinking about developing tools for therapeutics are wanting to steer away from a higher amount of uncertainty, right? You want to do a very chosen strategy for editing that you can predict. And you want it to be able to be exactly the change that you're looking for. But in plants, we have a tolerance for diversity, and it's something that I think we can really take advantage of. So what we've done is we've taken the human cell facility and taken existing tools and kind of started to see what we can do to work off of that base structure to push us more towards a diversification mixed base outcome. So systematically, by looking at the deaminase, glycosylation, various different aspects of the repair pathway from base editing, we can look at how do we push the entire repair pathway more towards a mixed base outcome as opposed to indels or just standard C to T editing. At the same time, we can look at other strategies that may look at inhibiting an undesired outcome at the same and as a result. And so this is a tool that's pretty early in advancement, so I can't really talk about the exact details of the tool, but I can show you what kind of the result of that is. And so from our human cells, we're able to see untreated, whereas we have a C, but when we introduce our C diversifier, we are able to see all 4 base types at that editing location. As you take this over to corn plants, we're able to see the same thing, but really what the exciting part to that is, at these sites, previously you would see just a couple of, you basically have one amino acid option. And we're now able to get a wider range of amino acid substitutions as a result of using this tool. And this then gives us a lot of ability to then start looking at diversity in a very localized sense at a protein, right? So we can basically take a active site of a protein, apply this tool, and start looking at diverse outcomes from an amino acid level and looking at phenotypes. So this is something that we think is a really powerful tool for us going forward. Okay, so I hope that was a good overview of kind of what it takes to kind of make a food company in the gene editing space. You know, we hope to be in this next few years, you'll start seeing some products that are a result of gene editing. And our hope is that that kind of helps consumers will be excited about it, and that we'll all be kind of excited about the different advancements to it that'll help people have a more healthy diet. And you know, really, it's all going to be driven by tool development. And so our hope is that over time, we'll help develop tools in a more broader space to kind of lead us towards a healthier diet. And so I would be happy to take any questions in the last couple of minutes.
Well, thanks for your patience. This worked earlier. And by the way, although the name says Sean Kaeppler on the control panel, this is Heidi Kaeppler, and I'm happy to be speaking today on our work in research in nanoparticle mediated approaches to genotype flexible germline editing in plants, and to acknowledge the co-PIs that are involved in this work with me. I've listed those here: Feng Zhang, who is in the Department of Genetics at the University of Wisconsin and also a member of the Wisconsin Institute for Discovery; Sarah Gong, and she's in the Department of Biomedical Engineering and Wisconsin Institute for Discovery; and also Chris Seha, he's in 2 departments of biomedical engineering and medical history and bioethics. And we pulled this group together for this research to pull nanoparticle related expertise that have been people working in mammalian systems and trying to apply those systems to the plant side.
Excuse me, you seem to be sharing your presenter slide, your presenter view, just so you know.
Oh, better?
Yeah.
Okay, well, we're going to speed along here. Sorry, again, this all worked earlier in the practice session. So anyway, current limitations that have been gone over in previous speakers already that we have in genome editing in plants and engineering include several components. And there's how do we get our components, whether it's for editing or engineering, into plant cells without killing or damaging the cells. Current systems such as the agro-based and biolistic systems and the regeneration systems needed for those are very genotype dependent across a number of species and genotypes. Trying to target cells that are capable of germline editing that are also regenerable and not genotype dependent. Current protocols kind of lack the ability to perform non-integrative type of editing. So right now, as in previous speakers have discussed, a lot of the engineering and editing work is done by integrating vectors expressing Cas9 and guide RNAs and selectable markers and so on, which can be a problem if you're not working in a grain crop and you want to segregate away that machinery, for example, if you're working in a clonal type of a crop. And also, again, the systems overall tend to have low efficiencies, complicated protocols or vectors involved, and of course, IP issues. So we initiated this work hoping to overcome some of those limitations with the nanoparticle mediated approach. And some of the advantages of those systems include the ease of the protocols in terms of physical delivery of the particles to our target cells and treatments. We can use nanoparticles to do both non-integrative and integrative genome editing. You have that ability to deliver more than one biomolecule simultaneously. So if you have several components you want to include in that particle, you can. And it also, the ability of the nanoparticles to enable local translocation of our cargo, whether it be to individual cells, organs, or tissues. Challenges in working with nanoparticles include the particle size limitations and transport limitations. How do you make a particle that retains and protects your cargo and that will release that cargo within the cell and within the right components? And then targeting and getting at those types of cells and tissues capable of genotype flexible regeneration of what we're interested in, and that's germline engineered and edited plants. So the overall research goal of this group and the project that we're on is to develop these nanoparticle-based systems to introduce our editing machinery into intact, so walled plant cells capable of regenerating germline edited plants in a genotype flexible manner. Our research challenges as a group and by group include particle design and assembly. So working on what are the best types, charges, size, surface chemistries to work with plant cells, confirmation assays of did we get cargo delivery, is it functional, whether it's genetic or epigenetic editing in the cells, and target and cell tissue types, what are the best types that we should be targeting, what are the treatment parameters for delivery that maintain cell health and regenerability, and then the regeneration protocols involved with that. The GON group is mainly aimed at the first challenge, and Juha Zhang's group mainly at the second, and my group mainly at the third. So in our first experiments, we've had this project going for just a little over a year or so now. Our first experiments just involved, can we get nanoparticles into target plant cells, and what's the size limitations on that? So for those experiments, we used FTSE labeled unimolecular micelle nanoparticles, not carrying any cargo at this point, but just as indicators of getting into plant cells, getting past the cell wall, because we're not targeting protoplasts in this work. There has been plenty of work on the human and mammalian side showing successful nanoparticle delivery and functional cargo delivery into non-walled types of cells, and that's what protoplasts are with plants. But the problem is ultimately protoplasts are a bad target for our objective of having genotype independent regeneration of germline edited plants, because regenerability from protoplasts is very genotype dependent, very species dependent, and ultimately wouldn't meet our goals. So could we get our nanoparticles into plant cells with intact cell walls? And so the first experiments, these images show where we have the overlay of FTSE expression of the FTSE labeled nanoparticles showing expression within plant cells, within the cytoplasm, chlorophyll back just to block out, here's where chlorophyll fluorescence is coming in, and then the merged systems showing that, yes, indeed, these test nanoparticles do get into the plant cells, do get into the cytoplasm, and we see, functional fluorescence. We also early on decided, let's take a look at uptake from root systems. How much transport of nanoparticles can we get throughout plants if we use the root system as our inoculation point? And so the pictures again show where we have FITC expression under FITC filter conditions in bright field, and what you're seeing are, yes, the nanoparticles get into in this case, Arabidopsis seedling roots and are transported throughout the stem and into the, in this case, it's showing seedling leaves, but also in more mature leaves. And unfortunately, in these images, we didn't dissect meristems, which is my main point of delivery that we'll be looking after, but we can show transport of the nanoparticles throughout the plants using this seedling type of assay. And so we looked at different sizes of particles in those experiments, mainly around 15 nanometers and 60 nanometers, and we did build some 140s. And so we found with those that we could get all of those sizes in, but the efficiency was better with the smaller size, which is pretty intuitive anyway when you think about pore limits and trying to get through cell walls and membranes and so on. So with that preliminary information, we then designed particles that were what we call stimuli responsive nanocage or nanocapsule particles to deliver our cargo. And what's shown in the picture here is we have the RNP assembled, and then the various processes of coating the nanoparticle, one to encourage uptake and or transport through the cell wall and uptake into the cell, either by hypothesized by endocytosis and or direct penetration of the cell membrane. And then these are also designed to protect that cargo in the process of being outside of the cell, inside of the cell, and if the particles are trapped in endosomes, they're designed to work with a proton pump type of strategy that will allow release of the particle from the endosome. And then within the cytosol, we have glutathione responsive chemistries on the nanocapsule that releases the coating and delivers the functional target, which is then transported into the nucleus. Now this is for like a single RNP molecule. If we also wanted to test whether we had the capability to deliver multiple RNPs within one nanoparticle. And so for this, we developed the redox responsive polymer-based polyplex platform. And so what the diagram shows here is that we have the RNPs encoded with this polymer-based type of coating with particular charges and overhang, and if we want to have targeting ligand attachments, and similarly through the same processes, and also being capable of endosome release and also the GSH responsiveness to only open up that capsule after the particle has reached the cytoplasm, and then eventual delivery into the nucleus. The sizes for the nanocages generally run in the range of 15 to 20 on average nanometers. The polyplexes are larger, and they can range in size, and you can almost estimate it from this picture here of anywhere from maybe 75 to 200 plus in size. Now, of course, smaller sizes we're showing, and others have shown, is better in terms of uptake efficiency. And so so we tested smaller versions of our nanoparticles, in this case where we had actually loaded cargo. And what you're seeing here is where we included in the nanoparticles an expression cassette for GFP DNA, and then we tried delivery into Arabidopsis leaves and also tobacco leaves. And so you see the top line across the board is showing GFP expression observed in controls versus different concentrations of nanoparticle treatments. And then we look under bright field and then merge images where we've also showed chloroplasts. I guess we have the chloroplast line missing. But what you can see is we have observed increased GFP expression at higher concentrations. What's not shown here though is that cell toxicity as you get into very high concentrations increases. And so, you know, the ability of those cells to live beyond the treatment for very many days is reduced. And if you're trying to regenerate a plant from those cells, then that would be a dead end. So, we're finding the balance of the best concentration versus and high delivery versus the toxicity. And so that's for these particular particles down in the first two concentration ranges. Similarly, we also did the same type of experiments with the GFP expression cassette containing nanoparticles in soybean. We first do our experiments kind of rather quickly in model plants like Arabidopsis and tobacco, and then we moved on into crop plants such as soybean and maize for the advanced experiments. And we get similar results also in soybean for leaf treatments. We then also wanted to test GFP mRNA delivery through the nanoparticles into plant cells, again, at first in models, Arabidopsis and Nocotiana. And similarly, we were able to successfully demonstrate and document expression of the GFP mRNA within the plant cells, and with highest expression observed 48 hours post-treatment. Then we also, the next natural step is, can we deliver RNPs into the intact leaf cells? And in this case, our goal was to knock out GFP expression within plants that were expressing GFP. And so we have on the left, the mock, so you see the GFP expression within the plant cells. And then with our Rep1 with our nanoparticle, this nanoparticle was in the 60 nm size range. And then Rep2, which is looking at a later time point, and then also a little bit of concentrations difference, but we can see, you know, kind of partial knockdown in Rep1 and complete knockdown in Rep2 of GFP expression by delivering the RNPs via these nanoparticles. So from that work, then we've been able to demonstrate cellular uptake of a number of nanoparticle sizes ranging from 15 to 140 nm into intact plant cells. We've been able to deliver functional DNA, RNA, and RNP cargoes into those intact cells. And we've noted, as others have, that the uptake of the smaller nanoparticles was determined to be more efficient. Also, we tested, and didn't have time to go through all of that today, but different surface charges and chemistries, nanoparticle concentrations, and these all can have various effects on nanoparticle uptake, cellular toxicity, and cargo delivery. So our current research and future research, so where we are and where we're going, is that we're in the process, basically Sarah Gong's lab is doing the fabrication and design of, and then we're doing the testing of the novel optimized biodegradable nanoparticle types. All of the types that we've worked with so far are biodegradable. They don't leave behind carbon nanotubes or gold or other non-degradable types of particles, and looking at those for delivery and uptake and expression of our cargoes. We're also moving those optimized nanoparticle treatments into targeting various cells and tissues capable of regenerating germline edited plants without being genotype dependent. And then and just down the line, we'll be testing when we want to look at applications where we have allele replacement in mind, or sequence insertion applications by homology directed repair, of trying the new simplex types.
Sorry, Heidi, you're
Yep.
out of time.
Okay, thank you.
Yep.
So sorry for the delays, and I'll
Yep.
take questions later.
Hello, everyone. Good afternoon. First, I would thank the organizer for invitation for me to participate and present my research in January, and also for introduction or moderating by Feng. And I'm going to talk about the genome editing in grass plants. And the first question is like, why the grass plants matter in our life? First, this is the plants belong to the Poaceae family or single family, and also including some of most important cereal crops like maize, rice, wheat, sorghum, etc. And also those cereal crops produce the world's most significant food source. And through years of research and accumulation, those plants have rich genetic resource and also intensive omics data. And because we face the world growing population, so we need to meet the demand for food. So those cereal crops or plants will hold the potential to meet the increased demand. And lastly, those plants provide the laboratory for development and application of biotechnology, including genome editing technologies. And for genome editing, this is the slides I use over again, and basically depend on several factors. One is programmable engineered nucleus, which can make genomic cuts at precise sites. And those cuts could trigger the endogenous cellular repair mechanism, and the repair could mainly through non-homologous joining or homology direct repair. So we can take advantage of this repair mechanism and create mutation at this cutting site. Basically, it's dependent on the double strand break. And also, most recently, thanks to David Liu's group work, and there's some base editing and prime editing, those technology do not need the double strand break, and also template DNA to repair or repair mechanisms. So that's including the base editing from A to G or C to T, and most recently use the prime editing can integrate the base the nucleotide change. For plant genome editing, usually it's different from animal, requires the regeneration of the plants to produce stable, transmissible edits into next generation could be used. So that's including the tissue culture and the transformation. And thanks work from many labs, and for the last several years, and huge or big collection of the plants have been subject to genome editing development and application. So it's including some of most all of the cereal crop plants. In our lab, basically we start work with the TALEN-based genome editing, and then expanded to CRISPR-Cas system editing. And for the CRISPR-Cas system, we built and adapted the platform of 2 vector systems. One, we use intermediate vector to design and construct guide RNA singly or in combination of multiple guide RNA for multiplex targeting. So with this gateway technology, we can mobilize the guide RNA intersect into the Cas construct. And in that case, we can generate single construct can produce both Cas protein and the guide RNA genes. And we also use the selection marker hygromycin or bioalphas resistant gene for the selection of the transformed plants. When we first built up the system years back, and we can demonstrate the system can produce very high efficient genome editing at the particular site in rice by target single gene. And then we use this as a tool and work on or systematically mutagenize a group of the sweet gene, especially expressed 15 of the sweet gene in rice. And we make single or combination of the genes mutation or knockouts, in this case, trying to characterize or trying to designate the gene function for those individual sweets. So through this systematic mutagenesis, we found several of the genes are indeed important for the biological process. For example, sweet 4, one knockout can lead to seed failing defect, and also sweet 11 and sweet 15 double knockout can also lead to seed failing problem. With individual sweet 11, sweet 13, and sweet 14 can lead to bacteria blight resistance, but those will maybe have some negative effect if we use for resistant application. Because CRISPR-Cas system can make multiple guide RNA to do the multiple legs targeting, we can use multiple guide RNA to do the large chromosomal fragment deletion. In this case, the biosynthesis gene of the diterpenoid could be deleted around 20 or 200 or 170 kb. One case I would like to demonstrate how use genome editing to make some real world problem solving or solving the real problem by use genome editing to modify some of the sweet gene promoter to make the plant more resistant to the bacteria blight of rice. So this is the working model through years of work from my lab and the Kaeppler lab or some other lab from the world come up with this model. The bacteria blight of rice caused by the Xanthomonas oryzae. Oryzae use a group of the tau effectors to activate the sweet gene in rice host. Once this gene activated, and the plant become more susceptible. There's some natural variations in the promoter of some of the sweet gene. For example, sweet 13 or sweet 11 contains the XC13 resistant gene. Those mutations happen in the promoter of the sweet 11 and makes the plant more resistant. So this is kind of group of the tau effector from different genetic background of the Xanthomonas oryzae and oryzae target different group of the rice gene. So the strategy come up with, if we can use multiplex gene targeting, generate multiple guide RNA, and target each individual guide RNA can simultaneously target their corresponding target site or in the promoter of the three sweet genes, so then we can create the promoter mutation, which is a different knockout of those individual genes. Then we may generate a single plant or group of plant will be more resistant or broad resistant to the bacteria pathogen collect from or happen in the different part of the world. So in this case, we come up with the 6 guide RNA based on the tRNA guide RNA platform. Each targets the tau effector target site. We call the EBE effector binding element of the sweet gene. For example, one guide RNA targets sweet 11 promoter corresponding to the tau effector of Xanthomonas oryzae PTHX1, so on with PTHX2 and some other tau effector target sweet 13, which contain group of the tau effector for sweet 13. So use this construct of the CRISPR-Cas9 system, we generate plants which contain a series of the mutations in different promoter of the sweet gene or combination of the mutation in different promoter of the sweet gene. For example, one of line contain 5 mutations in the at the tox site of the 3 promoters. So with those plants, this is the Kitake plants, is the model rice, which do not have much practical breeding value. With this proof of concept, we use Kitake to generate a series of the mutations or mutations in the promoter of the sweet and challenge those plants with different bacteria or Xanthomonas oryzae strains, as you can see here, and then we generate a series or different combination of the resistance to the bacteria blight of rice or different strain of the Xanthomonas oryzae. And we apply the similar the construct or similar technology to init rice, for example, IR64, which is widely adopted as the parent for lots of the init rice cultivars, and also Sharon Sap 1, which have tolerance to flood. So use the same technology and generate similar mutation in different promoter of the sweet, and those promoter mutations could have different phenotype of the disease resistance or susceptibility. Once the mutation has a good mutation or good mutations, they can generate very broad resistance. So this is the project funded by Gates and led by Wolf Frammer from Germany, and this is the group we call the Health Crops Group. And the future plan, we can develop and improve those init rice variety or expand it to other init rice variety and can provide the blight resistant rice variety to small farmers in Asia or Africa. Beyond this rice work, our lab collaborated with different collaborators and expanded the concept or the platform we use, this platform of 2 vector system, apply this genome edit or CRISPR-Cas9 system, particularly into different grass plants, for example, sorghum, wheat, maize, and switch grass. And basically this platform is, like I said, consists of the intermediate vector, which could be used to make the guide RNA and combine into the Cas9 system as in the single vector. And this system work for different genes in maize and have very good high efficiency for genome editing. And also for wheat, collaborate with one at South Dakota State University, we have the similar platform and can generate mutation or generate size-specific mutation in wheat. And also for sorghum, in collaborate with Jamie Yu at ISU, and we can use a similar platform and can generate mutations, for example, the flower locust gene. And last, I would appreciate the collaboration, the work effort from different group and also different people from my lab. I'll probably stop here. I go pretty fast.
My name is Arjun and I'm a plant synthetic biologist in the Voitis lab. Synthetic biology can allow us to make precise changes to plant signaling pathways to both elucidate biological mechanisms and engineer enhanced crops. However, actually implementing these techniques is a laborious, costly, and years-long process, thanks to the challenges of crop transformation. We've developed a new platform called Viparinama that uses RNA vectors to collapse these timelines from years to weeks. Come to my poster to learn more. My name is Arjun and I'm a...
Hi everyone, my name is Garrett Draper and I'm a PhD student in Dr. Dave Lagerspot's lab. The goal of our work is to generate a bottom-up model of malignant peripheral nerve sheath tumors associated with neurofibromatosis type 1 syndrome, or NF1, by combining CRISPR-Cas9 gene editing with induced pluripotent stem cells. These tumors arise from developing Schwann cells in deep nerves of NF1 patients that have been shown to harbor mutations in canonical tumor suppressor and epigenetic regulatory genes. By introducing these mutations along the developmental axis of Schwann cells derived from induced pluripotent stem cells, we hope to generate a novel iPS-derived preclinical tumor model that more accurately resembles MPNSTs collected from patients at the transcriptional level and that can be used to identify and validate potential druggable targets. If you want to hear more about this work, please come stop by my poster. Thanks. Hi, everyone. My name is Garrett Draper, and I'm a PhD student in Dr. Kaeppler.
Hello, everyone. My name is Jessica Woodley, and I am a research scientist in the CRISPR R&D group at IDT. Recently, we developed N-modified linear double-stranded DNA donors for use in HDR experiments. Through screening, we have identified several proprietary chemical modifications that both increase HDR rates and reduce unwanted homology-independent integration events at both the on-target and at off-target double-stranded DNA breaks, thus enabling us to provide high-quality HDR templates with higher yields that are otherwise costly and difficult to achieve with long single-stranded DNA donors without sacrificing on performance. If you'd like to hear more information, please come visit my poster to see data using these new modified donors. Thanks and have a great day. Hello.
Hello, I'm Ankit Padawan, a postdoctoral fellow here at Genome. I'm Heidi F Kaeppler in research technology, and we're working with Dr. Edgar and Dr. Scott's group at Mayo Clinic. Mitochondria has been a serious structure of independence that performs the vast variety of cellular functions. But when it goes to these mitochondrial disorders that are a constant affair for the clinician to treat. Despite six decades of existence, there have been very limited therapeutic options available to treat these kind of intelligence disorders. What we are doing here is we are working on gene-based therapeutics and how in the long term they can be served as a clinically viable therapeutic option to treat these kind of disorders. Harnessing the protein-encoded machinery of mitochondria, we are working on manipulating the mitochondrial DNA by new versions and improved versions of balance. We're also working on CPR proteins, which are cell-derived proteins of manipulating mitochondrial RNA. So if this is something that you're interested in and you would like to know more about our work on gene-based therapeutics for mitochondrial disorders, please visit our poster on building mitochondrial gene editing repertoire presented by myself and Keith on the back. Thank you.
Hi, my name is Gabriel Martinez, and I'm wrapping up my PhD at the Ecker Lab at Mayo Clinic. If you come to my poster, I would like to talk to you about how to make your genome engineering, be it for functional genomics applications or gene therapies, the most reliable it can be. Specifically, I want to talk to you about Menethu, a web software we developed that leverages our understanding of MMJ repair to find the double-strand break site in your gene of interest using whatever nuclease you want that maximizes your likelihood of getting a very consistent genotypic profile output. I would also want to talk to you about how this tool was validated and how it compares to other tools in the field. Thank you, and I hope to see you soon.